So this is the prep for lecture 7. For this one, we're going to just go through the steps of translation. So exactly what the mRNA is doing, how the ribosome moves along the RNA, and how the code is read and the amino acids are produced. The amino acid chain is produced. And there are just, this is a really good, this is a really good image from a Nature review. And it shows kind of the elongation step, which I think is very helpful. So you can see everything kind of laid out on this diagram. And these are the learning outcomes, so I won't read them to you because you can read them yourself. But these are the kind of things that we will be asking you about on quizzes and exams and so on, and in assignments and that sort of thing. And again, this is our conceptual framework, and we are here, and this is the final of the 3 lectures on this RNA to protein, the translation step. And so these are the factors that translation requires: the mature messenger RNA. In the case of eukaryotes, it's gone through processing and it's been transferred from the nucleus into the cytoplasm. For prokaryotes, the translation is taking place along with transcription, so it's a much more rapid process. A set of tRNAs, tRNA synthetases, which are the ones that recognize each tRNA and then put the correct amino acid on it. Ribosomes, little molecular machines. There are millions of them in cells. And they are the scaffold, and they are the place where the peptide bond is created, and some other proteins and so on that we're going to talk about now. So just to point out, the ribosome is quite an amazing little structure, and we draw it in this very simple way, but it's very complicated, and the average eukaryotic cell has somewhere between 1 and 10 million ribosomes in each cell. So they're very abundant in your body, in each of your cells. And even prokaryotes, bacterial cells, they're smaller cells, but they can have up to 15,000 ribosomes in a single cell. Right? So think of how small the cell is, and then that tells you how small the ribosomes are to pack, so much, so many of them into that small space. And they're not the only thing that's there. There's other material there as well. Okay, so tRNAs are a critically important part. We often draw the sort of cloverleaf structure, and the amino acid is attached to the 3' end, and the tRNA synthetases are the enzymes that attach. They recognize sequence of the tRNA, and then they bind amino acid to the correct position. And so this is how we draw it. If you were to look at it 3-dimensionally, it almost looks like a map of Africa in a way, although it's a little bit too skinny up at the top. And I like to just draw them this way. So I've got, this is the 3' end. And then the 5' end, and then the correct sequence to match the codon. And so just notice the 5' to 3'. So the anticodon is UGC, and anti refers to the fact that it's the reverse complement of the codon. So this is the messenger RNA, 5' to 3', and GCA is the codon. And then the anticodon you have to read in this direction, UGC, right? So you have to kind of write it this way and then flip it around, because I will ask you to give it to me in 5' to 3' order. Okay. And I think that might be all that you need to know. I'm not so concerned with you sort of remembering like all the names of each of these and so on. Just the idea that there are these series of stem loop structures which come from the RNA folding back and base pairing with itself, that the 3' hydroxyl, the 3' most part sticks out a little bit more, and that's where the amino acid is attached. And then the anti-parallel nature of the anticodons and the codons. Now, there are 61 different codons, but there are not 61 different tRNA genes. And the reason is that tRNAs have what is called the wobble position. And what that means is that on the 5' end, it's partly just physical. You've got your messenger RNA, and then you've got the sort of the stem loop structure, and the way the amino acids are, the way the nucleotides are arranged, they can't sit completely flat on the messenger RNA. And so one part is kind of pulled upwards just a little bit, just slightly. And what that means is the binding is tighter between the first 2 nucleotides of the codon and the second 2 of the anticodon, but the first nucleotide of the codon and the last of the anticodon, there is this sort of pulling upwards so that the nucleotide is sticking out at an angle a little bit. So it's able to bind with either a C or a U. And that's useful because it means that you have one gene for this tRNA, but it can recognize 2 different codons and bring the correct amino acid. So it's a kind of efficiency, right? So this particular tRNA can bind UCC and UCU. And so this is an example of the serine ones. And there are 3 different sets of tRNA genes, but there are 6 different codons that could be recognized. And this just kind of shows you the anticodon is AGG that can bind to, when it says plus wobble, what it means is that the AGG is able to bind both this codon and this codon. And just note this is 3', 5', and this is 5', 3', right? And so just remember that the 5' most nucleotide of the anticodon, there's a little bit of upward pull just because the RNA can't sit flat on the messenger RNA, and so that means it can bind 2 different nucleotides. I don't care if I'm not interested in you necessarily memorizing this, but if you remembered one of them as an example, the commonest thing to ask directly about this would be, just please explain why we have 61 codons, but there's fewer than 61 tRNA genes. What's that about? What's causing that? And this would be the explanation. Okay, so just like the other processes, there is initiation, there is elongation, and then there is extension. And the initiation is quite interesting because it happens in stepwise process. So there are factors that start the assembly of a special tRNA. This little I means its initiator. So the first amino acid that's put on is methionine. It recognizes AUG, which is the start codon. But so that has to sit down on the DNA, the messenger RNA, and the 30S ribosomal RNA subunit, not ribosomal RNA, but ribosome subunit composed of RNA and protein. And this is the small subunit, okay? And so there's a specific sequence called the Shine-Delgarno sequence. It's named after the 2 people that discovered it and worked on it together. And it's near the 5' end. So you've got your messenger RNA. And in prokaryotes, there's a little sequence right at the 5' end that is where the ribosome binds. and then right beside it is the AUG, and the special tRNA binds there. And when all of that is set up properly, then the rest of the ribosome binds on there. And so the ribosome assembles kind of around the RNA. So the small subunit first, and then the larger subunit comes afterwards. So the ribosome doesn't assemble and then the messenger RNA is fed into it. It just builds itself right around that messenger RNA. And so this is the Shine-Dalgarno sequence. And so part of the RNA in the small subunit is called the 16S RNA. This is a measure of the size of it. But just remember that one of the RNA components of the small subunit has the same reverse complementarity to the messenger RNA. So the messenger RNA is read 5' to 3' like this, and then the 16S RNA 5' to 3' this way, and they bind to each other through complementary base pairing. And then basically from the position where the small subunit has bound, the tRNA with the initiator formal methionine. It's a specially modified methionine. It usually gets cut off after the protein is finished, but that tRNA is going to find the first AUG from the Shine-Dalgarno sequence. And that's how the reading frame is set. That's going to be your start codon, and then everything is read in 3s after that, right? So there's two things that are happening. We're starting the whole process, but we're also setting how the RNA is going to be read, and this is called the reading frame. Okay, so this is a computer model. This is a schematic, and the idea is you have the A site, you have the P site, and you have the E site. And so this isn't exactly what it means, but I think of the A site as the arrival center. That's where the new RNA is going to come into. The P site is where the peptide bond is being made, and the E is the exit site when the tRNA is actually released. Okay, and so what we're going to see is the ribosome sits there, and a new tRNA comes into the A site. The whole ribosome kind of shifts along, and when it does that, there's conformational, so there's slight shape changes that causes the release of whatever tRNA is still at this site. And we'll go through the steps in more detail in a moment. But so think about each of these as where different events are taking place. Arrival and correct recognition and binding of the anticodon to the codon, and then the place where through shape change, which I'll explain in a minute, the peptide bond is made. And then this is now empty, and the ribosome shifts. A new tRNA comes in, and now this one is in the P site with its peptide chain on it. This one is empty, and it gets released, and it's just repeated over and over again. And this is the diagram that is helpful. So here is our site. We have our tRNA with our peptide chain on it, and here is the empty tRNA. So the ribosome shifts along the RNA, and when it does this, a new transfer RNA with its amino acid is bound to EF, elongation factor 2, and it's GTP. GTP is a ribonucleotide, but it's going to be hydrolyzed to make energy, and that energy is going to allow for the formation of well, first of all, for the recognition of the tRNA on the right position, and it's set in just right, and then the next one will catalyze that peptide bond formation. Okay, so here comes our tRNA. If this is if this matches, then everything is good. um, the the elongation factor GTP is hydrolyzed to make GDP, and it gets released, and that leaves the tRNA in that position. Now, it's possible that this isn't a correct match, and if it isn't a correct match, that tRNA gets released, and it has to start over again, find another EF2 GTP, bind to it, and so on, and a new tRNA comes in. But for this purpose, let's assume this is the correct amino acid, or sorry, the correct anticodon. And then when both of these are bound, there's a little bit of a shifting, and you can see it here, sort of. It kind of shifts these two tRNAs close to each other so that this amino acid chain can be attached to that one. And it happens because they're very close to each other. Okay, and the RNA component of the ribosomes appears to be the part that catalyzes or allows for this reaction to take place. Okay, so once that happens, once the chain is transferred to the new tRNA, then that again changes conformation, so they now separate again, and now you've got the empty one sitting here, and elongation factor GTP, comes in, and it kind of binds the nucleotides next to, it wants to bind in this A site, and so it's kind of coming in, and I think of it, this is the wrong way to think of it, but I think of it as shoving these guys along. But in fact, what's happening is it's coming in, and the ribosome is shifting over, and it's basically binding here and causing the ribosome to shift over, and the GTP is hydrolyzed for that to happen. And so once the GTP is hydrolyzed, this is released. But now what's happened is the ribosome is shifted along, and it's made this A site empty. Okay, so everything is now, because the ribosome is moved, now the thing that was the chain and the tRNA that was in the A site is now in the P site. The empty one that was left over at the P site is now in the E site. And the GDP is released, so we've got this empty position here. And then next comes the next amino acid. So the next amino acid is coming in on this tRNA with EF2 GTP. It's going to bind in here, and when it does that, it changes the shape a little bit, and that pushes out the empty tRNA, and repeat and repeat and repeat. So this is the elongation process. You might want to talk it through a few times. And I think I haven't looked at what's in our codon site yet, but I will make sure that we have some little animations posted there that will help to visualize the process. But trying to draw it out, trying to think it through, talk it through as you go through the diagram and so on. These are really good ways of trying to understand that step. And so these steps are repeated multiple times, of course, for each new amino acid that gets added. And eventually, at the beginning you don't see it so well, but eventually the amino acid chain starts to stick out from the ribosome as it's moving. This process occurs really quickly. And the protein starts to fold. It'll start to take on those pleated sheets. It'll start to take on alpha helices and so on before the protein is complete, and then folding over and creating the secondary and tertiary structures. And so the protein is folding up and adopting its proper shape as the translation process is taking place. In prokaryotes, when a stop codon is reached, there's no tRNA that binds that. unless there's been a mutation, which we'll talk about in a minute. But instead, there's two release factors. Release factor 1 recognizes these two codons. Release factor 2 recognizes these two. So notice that both of them recognize this one. And so this is not base pairing anymore. This is a protein, and the protein happens to have a shape that matches, one of these stop codons. And when that happens, the release factor binds in there, and then it triggers the release of the amino acid sequence, and that is released, and it sets up the rest of the process to terminate. Okay, so it causes a cleavage, right, and release of the polypeptide chain, as I said. And then there's another factor called release factor 3, and that basically causes release factor 1 or 2, whichever one is bound here, to move from the ribosome messenger RNA, and the ribosome subunits also fall apart, and the tRNAs are all released. They no longer, neither one of them has an amino acid chain attached to them. So release factor 3 stimulates, once the peptide is cleaved and released, then release factor 3 stimulates sort of the release of the RF factors and the falling apart of the ribosome, and they disassemble, they get reused. Again, the tRNAs get recharged. The protein folds up and goes on its way doing its job. It gets transported to where it needs to be, or is cytoplasmic, or goes back to the nucleus to transcribe genes, whatever that protein is doing. And the two ribosomal subunits are able to reassemble on a new messenger RNA, and the whole process can begin again. Now, nonsense mutations, as I talked about, this is from lecture 6. But nonsense mutations are interesting because they cause truncated proteins. And Sidney Brenner, who was an incredibly famous powerhouse in the world of genetics, wanted to find out if you could find one mutation that suppresses another. So if you have a mutant phenotype, sometimes you can mutate another gene so that the protein will now bind with the mutated protein and allow the function to proceed, allow the proteins to do whatever the job is that they're supposed to do. It's kind of interesting. And so he kind of pioneered this, and he found suppressor mutations of nonsense mutations. And so, for example, if you had UCG, a codon for serine, and that had been mutated to a UAG, that's a stop codon. You would get premature termination of the translation because the release factors will come in there and stop translation and release everything. And your protein is truncated. That means it's been cut. It's shorter than normal. And these mutations were called Amber, after his daughter, or the daughter of one of his students, I can't remember which. And then because amber is a kind of like a precious stone in a way, then they called the next one UGA opal and UAA ochre. And so these are the stop codon mutations. These are nonsense mutations. So an amber mutation is something like this: a serine codon changed to a stop codon. UGA is another, UCA changed to UGA. That's called opal, and UAU changed to UAA. That's called ochre mutation. And it turns out you can actually isolate mutations in other genes. And when you find nonsense suppressors, what has happened is the stop codon is no longer read as a stop codon. It's actually allows an amino acid to be put in, to the peptide chain. It might not be the correct one, but it's an amino acid. And then you get the continuation of the production of the protein, and you can go back to wild type, which is very interesting. And it turns out these are mostly mutations in tRNAs. Now, if you want to test yourself for your understanding, you could stop the tape at this point and ask yourself, where in the tRNA would we make a mutation that would allow it to read a stop codon and then put in an amino acid instead of the release factors coming in and binding. And it turns out, as you might have guessed, it's a change in the actual anticodon. So the tyrosine tRNA, the anticodon is AUG, but if it's changed to AUC, this is 3' to 5'. So if there's a change, that means that this tRNA will bind to a stop codon and add a tyrosine to it, and that allows, the next tRNA to come in and, put its amino acid, and then the next one, and so on. And so now, instead of having a truncated protein that stops right here with only 3, or whatever this is, 1, 2, 3, so with only 6 amino acids, now you can get the rest of the protein made. And as long as the amino acid that's put in by mistake it doesn't hugely get in the way of the function of the protein, then it proceeds normally. Okay, so that's a really quick run through the translation process. There's going to be more details on the resources and things that I've posted, but this is the level at which I hope you'll be able to answer questions. And our next topic starts to get more into heredity, how genetic information gets passed to the next generation. And so we'll do a quick review of mitosis, which is the regular type of cell division where cells make identical copies of themselves, and then we'll follow that with gamete formation, which happens through a special form of mitosis called meiosis.